Hello dear students, today we are presenting a special program on Peziza, important features and life cycle. The objectives of today's lecture are taxonomic description of Peziza, structure of Mycelium of Peziza, reproduction in Peziza, both asexual and sexual, life cycle of Peziza, economic importance of Peziza. Peziza, with over 100 species, belongs to family Pezizaceae, order Pezizales, class Ascomycetes, and division A. Mustigomycota. Division A. Mustigomycota is characterized by having absorptive type of nutrition and absence of motile spores in its member species. Dear students, fungi in class Ascomycetes have septate hyphae and bear specialized spores called ascospores, produced endogenously in specialized sporangium, known as ascus. Ainsworth, the famous botanist, raised the class Ascomycetes to the subdivision category as Ascomycotina and subsequently recognized 6 classes within it, with Discomycetes as one of them. Discomycetes containing the genus Peziza constitutes the group of fungi having apothecium as ascocarp, that is, the fruiting body. The apothecial ascocarps produced have an exposed hymenium at maturity. Pezizales is a large order that contains the species commonly called operculate Discomycetes as well as derived hypogeous forms that have evanescent asci with ascospores spread by mycophagy. Family Pezizaceae is characterized by having fleshy or brittle, sessile or feebly stalked and usually cup or disc-shaped apothecia. Peziza is the largest genus in the order Pezizales. It includes the fungal species, whereas asci line is the inner or upper surface of a cup-shaped fruiting body. The students, the ascus opens at maturity and discharges its spores into the air. Most members of this genus are difficult to identify as they look outwardly very similar. So identification and verification of microscopic features is usually required for identification. The spore ornamentation, which is visible when stained with cotton blue, is used in the identification of species. In common with other Ascomycetes, the upper surface of the fungus has a layer of cylindrical spore-producing cells called asci, from which the ascospores are forcibly discharged. Like a mushroom, Peziza produces above-ground heterokaryotic reproductive structures that come from an underground mycelium. One way that Peziza differs from mushrooms is that it produces spores on top of its cup, not underneath like a mushroom. Like species delimitations, intergeneric and infrageneric relationships of Peziza are debatable. Recent molecular analysis of rDNA and many other genes suggest that the genus is paraphyletic. Preliminary analysis based on nuclear large subunit rDNA sequences from a broad sampling of taxa within Peziza as well as from related genera suggests that all other genera in the Pezizaceae are nested within Peziza. Some species of Peziza appear to be more closely related to the species in other genera within the family than they are to some other species within genus Peziza. This infers that either more species would need to be drawn into Peziza or Peziza will need to be broken up into a number of genera. The morphologically deviating species Peziza girardi appears to be the basal lineage in the Pezizaceae. The lineages broadly correlate with spore morphology and pigmentation. Dear students, morphological characters previously used to delimit species within the Peziza species complex, such as color variation of the apothecia, presence or absence of a stipe, stratified or non-stratified medullary accipul or thickness of the accipular layers, cell types in the outermost accipul, and monoliform versus filiform paraphysis are not correlated with the subgroup supported by ITS analysis and appear to be plastic. Further, the substrate and habitat have been much emphasized in the taxonomy of Peziza, but the populations on a diverse array of substrates may be closely related or indeed conspecific. Still, Students, certain morphological characters are used to distinguish different species. For example, Peziza Peziza is recognized by its flattened purple cup-like fruiting bodies. Dear students, a combination of characters makes Peziza vesiculosa relatively easy to identify. Most obvious is its preference for fruiting in clusters on horse dung or decayed straw. Peziza vesiculosa is additionally distinguished by an urn-shaped cup with a margin incurved even at maturity, and a wrinkled yellowish-brown hymeneal inner surface and a pale outer surface with minute pustules. Fruiting body in Peziza vesiculosa is 2 to 5 centimeters broad, sessile, often contorted when clustered. Margin is incurved, remaining so with age, at times erodes or cracks with age. The hymeneum or the inner surface is light brown, pale yellow-brown to medium brown in color, frequently convoluted or wrinkled. The outer surface of hymeneum is granulose to furraceous, sometimes indistinctly so when weathered, tan, pale buff to nearly white. Peziza domiciliana is a brownish cup fungus, while as Peziza cerea is a yellow-gray to beige fungus, internally less than 5 centimeters across with a granular or brittle flesh. The spores in Peziza cerea are smooth or elliptical, white-colored or creamy or yellowish. The cup exterior is white in color. Its upper surface at maturity is usually somewhat wrinkled near the center, a whitish and minutely fuzzy undersurface, round, cup-like shape when young, and a flattened, irregular shape when mature. Peziza repanda can be identified by its growth on rotten wood or wood chips. Its brown upper surface at maturity that is usually somewhat wrinkled near the center, a whitish and minutely fuzzy undersurface, a round cup-like shape when young, and a flattened irregular shape when mature. Elliptical spores that lack oil droplets. Peziza depressa has a fruiting body about 20 to 40 millimeters, irregularly cup to saucer shaped. The fruiting body has short stipe and its inner surface is smooth, purple to chestnut brown. The outer surface is almost smooth, brighter than the inner surface. The fruiting body exuding a watery juice when cut. Peziza domiciliana is a brownish cup fungus. It is often found to grow through cracks in concrete. It is also found in sandstone and in crumbling building remains, as well as in carpets, plasters, shower stalls, and so on. It is not only the species that could conceivably grow in such settings, but it is the species that is usually found indoors or in concrete. Also, dear students, the closely related Peziza praetervisa is also violet colored and prefers growing on burned ground. In general, Peziza praetervisa is more purple rather than violet colored, like Peziza violacea. However, fruiting body color can vary depending on humidity and other factors, so they are more reliably distinguished microscopically. Peziza praetervisa has a rough, not so smooth spores with 2 polar oil drops. Fruiting bodies in Peziza praetervisa resemble flattened cups with a diameter of up to 3 centimeters. The inner spore-bearing surface, the hymeneum, is purple-brown but lightens to brown with age. Also, one important thing that you have to remember is that the exterior surface is pale purple and scurfy. This species is sessile and does not have a stipe. The flesh is thin and mauve colored. Spores in Peziza praetervisa are elliptical, hyaline, with small fine warts on the surface and have dimensions of 12 to 14 by 7 to 8 micrometers. They are bigatulate, containing 2 oil drops at either end of the spores. Dear friends, the asci are 250 to 300 by 10 to 12 micrometers. Peziza bedioconfusa has cups of 3 to 10 centimeters across, deeply cup-shaped, becoming irregular. Inner surface is dull reddish brown. The outer surface is similar but roughened or unpolished. There is no stipe in this species. The asci are about 140 by 12 micrometer in size. The spores are ellipsoid, finely wadded, containing 2 oil drops of 17 to 21 by 8 to 10 micrometer size. The mycelium, dear students, is frequently perennial and consists of a dense network of branched septate hyphae. The cells are uninucleate, that's containing only one nucleus. The hyphae ramify within the substratum and form a complex system which extracts nutrients from the substratum. The apothecia are aerial extensions of the subterranean mycelial network. The mycelium is frequently perennial and consists of a dense network of branched septate hyphae. The cells are uninucleate. The hyphae ramify within the substratum and form a complex system which extracts nutrients from the substratum. The apothecia are aerial extensions of the subterranean mycelial network. They are semi-immersed in Peziza ammophila, while in certain species they are flattened. The size of apothecium varies from 1 to several centimeters. Similarly, they vary in color. For example, they are bright orange in Peziza aurantia. The exposed hymenium, which lines the interior of the apothecium, consists of elongated asci and paraphyses. The primary wall in ascospores is devoid of chitin and is composed of alpha-1,3 glucons. Barium permanganate treatment does not indicate appreciable amounts of protein and lipids in it. Ultrastructural data confirm light microscopic studies that show an expansion of the investing membrane within which further wall deposition occurs. One more important thing to remember is that the coalescing of cytoplasmic vesicles with the investing membrane creates a large vacuole, the perisporic sac, around the spore. Simultaneous with the formation of the perisporic sac, osmiophilic lumps are deposited on the primary spore wall. Dear students, the osmiophilic lumps increase, coalesce and form the initial epispore. The epispore consists of zones of bands of varying electron opacity whose staining properties indicate complex carbohydrates. Dear students, the further deposition of material from the perisporic area results in the formation of spore ornaments made up of fibrillar strands. The staining reactions indicate that spore ornaments consist of a matrix of lipids, proteins, glycoproteins and chitin. One more important thing to remember is that the ascospores are released from the apothecium generally in the moist environmental conditions. On germination, the ascospores give rise to a germ tube which develops into a new mycelium. Well, the asexual reproduction is absent in most of the species of Peziza, but a few species produce conidia and chlamydospores. At the time of formation of conidia, some hyphae grow upwards and act as conidiophores. The conidiophores, students, you must remember, are long, cylindrical, erect and septate. The tip of the conidiophores swells to form a vesicle. Conidia are formed on the surface of the vesicle. They are hyaline, elliptical and thin-walled. They germinate on the suitable substratum and produce new mycelia. Chlamydospores are thick-walled resting cells formed intercalary on the hyphae either singly or in chains. In favorable conditions, they germinate and produce germ tubes which eventually grow into new mycelia. Definite sex organs, that is antheridium and ascogonium, are not formed in Peziza. Sexual reproduction takes place by somatogamous copulation. Some hyphal branches in the mass of hyphae bear dense protoplasm and the two adjacent cells of these hyphae fuse to form a dikaryotic cell. However, in certain cases, one must remember that the autogamous pairing takes place. Cell division in the dikaryotic cell occurs in such a way that the resulting cells are dikaryotic and form an ascogenous hypha. The dikaryotic hyphae grow together to form a reproductive structure called an ascocarp. The two nuclei in the terminal cell fuse and become a diploid nucleus. Also, the ascus then elongates and the diploid nucleus by meiosis forms 4 haploid nuclei. Each haploid nucleus divides again by mitosis, resulting in a total of 8 haploid nuclei which metamorphose into haploid ascospores. On their germination, they form the primary mycelia which can produce microscopic hyphal mass. It represents the haploid phase of the fungus. Thus, in the life cycle of Peziza, there is a long haploid phase followed by a short dikaryotic phase and a transitory diploid phase. The mature apothecium is a cup-shaped, sessile or subsessile structure 1 to 10 centimeters in diameter. They may be whitish yellow as in Peziza aurantia or bright red or gray in color, for example in Peziza vesiculosus. In Peziza arverensis, they are solitary, sessile, cupulate, extremely whitish and nearly smooth. Their margins are slightly wavy, 3 to 10 centimeters in diameter and often irregular in outline, but eventually spreading. Dear students, you must remember that the hymenium is concave, even wrinkled or furrowed, glabrous light brown to chestnut brown in Peziza arverensis. The exterior surface is white to ash gray, finely tomentose. The asci are arranged in parallel rows within the apothecium. In apothecia receiving light from above, the asci at the base of the cup are straight, while those on the sides are curved towards the light. Importantly, this orientation of the asci ensures smooth upward discharge of ascospores. The asci have a distinct amyloid ring at their apex, which stains blue with iodine. Ascospores are large, elliptical and are discharged violently and in one lot. On the basis of ascospore characteristics, the species of Peziza fall into two series. 1. Those producing smooth-walled spores without oil drops, and those producing spores ornamented by warts or ridges and usually containing 2 large oil drops. Dear students, some species of the second series yield a colored fluid from the flesh of their ascoscarp. The ascospores on germination at the suitable substratum give rise to a haploid mycelium of uninucleate cells. Also, a longitudinal section of the apothecium shows the following regions. Number 1, hymenium. It is the innermost region of the apothecium and contains asci, ascospores and paraphyses. Paraphyses are sterile cells often with swollen tips and are at high turgor pressure and are placed at right angle to the surface of the apothecium. Tips of the paraphyses are very tightly together at the surface of the hymenium and create a barrier, the epithelium. The exposed hymenium, which lines the interior of the cup, consists of elongated asci exhibiting various stages of development and paraphyses. Both the asci and paraphyses, you must remember, are phototropic and their tips are always bent towards incident light. To disperse spores, asci push between the paraphyses from below, shoot off their spores and then collapse. Number 2 is the subhymenium. The middle region of apothecium is known as subhymenium or hypothecium. It's composed of thin-walled and light-colored hyphae which run parallel to the surface of the hymenium. The asci and paraphyses arise from these hyphae. Number 3 is the excipulum. It is the outermost region of the apothecium composed of sterile hyphae. Now moving on to the economic importance of Peziza. In general, the species of Peziza are with no apparent use and there is no concrete evidence about their edibility. However, the species found growing on soil have a tendency to increase soil fertility. The species growing on wood logs lead to deterioration of quality of timber. The species growing indoors are potential pollutants. A unique hemaggulating lectin has been isolated from Peziza arverensis. This lectin determined to be a single chain protein and is of biotechnological importance. That is all we had for you in this edition of the classes of botany. Hope you enjoyed the lecture.